next keynote speaker is Dr. Jagadananda Ghosh और मैं आपको बताना चाहूँगा Dr. Ghosh is a senior scientist at the Henry Ford Health System in Detroit, Michigan. He is working on signal transduction and developmental therapeutics for the last 20 years using prostate, lung and kidney cancer as model systems. He has many years of experience in the area of genetic analysis, signal transduction, drug screening, and mouse models in prostate cancer that makes him uniquely suited to lead and carry out the work in this proposal. Please welcome Dr. Jagadananda Ghosh. Can we have the huge round of applause?
Hi everybody. I'd like to thank the organizers particularly for giving me this opportunity to visit my motherland, number one, and also for giving me opportunity to present some of the work that we are doing lately. So from the title you can imagine that it's about cancer drug resistance. Now, the drug resistance was not a problem 50 years ago because there were hardly any treatment. And now we have chemotherapy, radiotherapy, even immunotherapy, targeted therapy, a lot of therapies, but still cancer is the winner and we are not. And in many cases you can see the burden of the disease, it is very debilitating, and eventually we lose, meaning the cancer kills the patient. Very rare cases, there are some lucky patients, they might say like, well, I'm cancer free, very rare cases, or with the cancer they live for long enough and say eventually goodbye, like, well, the therapy really helped me. But that is not the point that we should be happy or satisfied about. Our expectation is when the regular treatment fails, the disease resists, the disease become resistant. Can you do something? So that's the topic for today's discussion. So cancer drug resistance, and there is a new mechanism, relatively new mechanism, that is cancer cellular transdifferentiation. When we were students, we used to hear a lot about cancer drug resistance because of drug efflux pump, the drug metabolism, the mutation, target modification, and now we are at the fourth dimension of the cancer drug resistance. That is the cellular transdifferentiation. And I'll show you some of the data to show the role of this molecule. This gene that particularly we found in prostate, it's not new, we didn't discover, but we found the new gene in very advanced stage lethal prostate cancer. So Tribbles 2, please remember the name. And we take prostate as an example because it's a very complicated disease, particularly because of its strategic location just below the bladder, which we are going to. Couple of slides. Yeah, here. So just below the bladder, urinary bladder. So a prostate, the problem happens in the prostate. The immediate symptoms is urination. So meaning urine cannot pass because it compresses the urethra, where the urinary pipe. So the urine builds up in the bladder. That eventually creates a problem in the kidney. So the prostate cancer is a disease of 3 in 1. A prostate problem creates a bladder problem, creates a kidney problem. Highly prevalent in US, you can see 1 in 8. There's a random statistics. 34,000 deaths per year. So you can imagine by the time I started talking, by the time I have 20 minutes, so by the time I'll finish my talk, this disease will take at least 1 life in US only. Considering the whole world, it may be several. So that's the magnitude of the problem. Now, for advanced prostate cancer, most of the deaths happen due to ERPC, enzalutamide resistant prostate cancer type disease. Now, prostate cancer starts as an androgen-driven disease. Androgens are produced by these glands. Work on the prostate tumor cells. It triggers androgen receptor-mediated signaling, some gene expression, and eventually promotes cell division, meaning that the cell starts proliferating. And if the proliferation never stops, it makes a tumor that eventually metastasizes. A lot of complication happens. Now, fortunately, we have some treatments you can block androgen production, or you can straight go to the androgen receptor, and there are good drugs. These are good drugs, usually enzalutamide, apalutamide, darolutamide in the middle, you can see. But still, enzalutamide is the most popular because almost every prostate cancer patient with advanced disease gets this drug. And eventually resistance happens, and that's the killer version of the disease. And the enzalutamide is currently used in the clinic at this dose, 160 milligram per day for 12 weeks. So for finding a new treatment, what we needed to do, finding a critical target. If you know a target, you can do something. So what we did, we developed a model. We wanted a cell culture model to go deep into the molecular mechanism. Other models can be a tumor analysis, another model RNA analysis, a lot of analysis you can imagine, but our interest was to go to molecular mechanism-based target. So we needed a model. What will be the model? Our model was the cell culture. What we did, we gradually treated cells with increasing doses of enzalutamide. First 10 micromolar for 4 weeks, then raised to 20 micromolar for another 4 weeks, 30 micromolar for another 4 weeks. Now you can ask like, oh, why 30 micromolar is the magic number you use the drug to make the cells resistant? Now 30 micromolar is the blood level of the drug in a standard clinical treatment to the patients. So thousands of patients were sampled, and average blood level was 30 micromolar. So we thought that, okay, 30 micromolar drug resistant cells will be our really ERPC cells. So eventually we have two sets of cell lines. The parental cell line, enzalutamide sensitive, the resistant cell lines after 12 weeks, because that's the way the patients are treated in the clinic. Gene expression, whole genome, 34,000 genes, and eventually found in D, you can see TRIB2, Tribbles 2 in short form. Now the genes are, these are planar, the red ones on the left side, these are the genes, those are overexpressed in the resistant cells, and the green ones are underexpressed when they're going down regulated after the cells become resistant. And you can easily see that Tribbles 2 is at the top. So when we found this gene by gene expression array, we have to confirm it by other tests. So the first test we did is A, RT-PCR, by RT-PCR mechanism. By the time we have two sets of cell lines, LN cap is enza resistant, PCR2B resistant. Both of them show that increased expression of mRNA when the cells become resistant. Then we move forward to do Western blot. By the time we have three sets of cell lines, LN cap ENR, PCA2B ENR, LFAC4 ENR, those are the standard prostate cancer cell line. Those we made resistant by the same mechanism, giving increasing doses of the drug treatment. And in the Western blot, you can see Tribbles is up in all three cases. So these are in vitro. So in vivo, you're seeing that these are PDX tumors, patient tumors implanted in mice, treated or untreated with the drug, enzalutamide treated. You can see with the drug treated, there is a crossover expression of Tribbles 2. And the right side is the quantitation, right? Tumor after tumor, finally, these are human patient samples collected from the patients who have just died. So it's called warm autopsy. Patient died, but the body is still warm. So sometimes the patients donate their body for research purpose. So these tumors were collected from those patients. So you can see that the treated patients have grossly overexpression of Tribbles 2, compared to the untreated ones. And the right side, the green and red, you are seeing the quantitation of the measurement of intensity of staining. You take the average. So you have each dot is a patient sample. So up to this point, our conclusion is Tribbles 2 gene is overexpressed in ERPC cells and in ERPC tumors. Now the question is, well, does it play any role? If it works better, it's good, it's a marker. But what are we going to do with a new marker? Unless it has a good role. So that was the question that we wanted to answer, like what is the role of, what is this doing in the cancer? So as of today, there is no commercially available good Tribbles 2 inhibitor. So what are you going to do? shRNA. So you knock out shRNA, Tribbles 2 with shRNA, the phospho-AKT goes down, FOXO3 goes up, that's a tumor structure. On the B, you can see the cells are largely affected. These cells are dying, meaning These cells, the resistant cells, they have, not only they have Tribbles 2 overexpressed, they depend on Tribbles 2 for their survival. In C, you are seeing the quantitation, the cancer cells are specifically affected, but BPH1 in benign prostate hyperplasia, that doesn't have much Tribbles 2 and they're not dependent, so they're not dying. So it is a little bit cancer specific. And in vivo, you can see the mice, they have tumors. If you knock down their Tribbles 2 by FANA, fluoride modified antisense oligonucleotide, in vitro you are seeing the document and the cell survival on the E. In the F is the real example. The cells, the resistant cells make good sized tumor, but in that tumor, if you inhibit their Tribbles 2 with FANA oligo, there is a really good amount of difference, meaning it decreases the tumor growth if you block their Tribbles 2. So that's a good news for us. The other way you can test the importance of Tribbles 2 is expressing Tribbles 2 gene, overexpressing Tribbles 2, not by drug treatment, but just transfecting the cells with gene itself. So we have a construct GFP level Tribbles 2. You can see a big strong band, LN cap Tribbles 2, PCA2B Tribbles 2. Strong band, these cells are AKT now high, BCLXL, those are molecular marker. On the right, you can see LN cap parental cells and the Tribbles 2 transfected cell. You can easily see the transfected cells have bigger colonies and more numbers. And on the right bar is the quantitation. These cells are more invasive also. In vitro chamber, these cells, Tribbles 2 overexpressed cells, invade more. And that is comparable to with metastatic ability. The real test is here. In A, what you are seeing, that the green bars, LN cap cells, parental cells, they're sensitive to enzalutamide, 10, 20, 30 mark model, increase the dose, the cell viability going low and low and low. But the red bars, the Tribbles 2, when you overexpress, just one gene overexpressed, they're completely resistant to enzalutamide treatment. So up to 30 micromolar, you barely see any change. Now, these cells are also resistant to apalutamide, darolutamide, and abiraterone. So all the second generation androgen signaling blockers will be ineffective if this gene gets a chance to overexpress by some mechanism. We don't know the complete story yet, it's a very fresh data. In B, these cells, they make colonies, LN cap cells make colonies inhibited by enzalutamide. On the right side, you see the LN cap Tribbles 2 bigger colonies, and there is hardly any inhibition with 30 micromolar enzalutamide treatment. And in mice, we have LN cap and LN cap Tribbles 2 implanted on the same mice, one side, LN cap one side, the resistant cell. With enzalutamide treatment of the same mice, LN cap tumors decrease the growth, but Tribbles 2 overexpressed tumors, there are hardly any change. You can see on the quantitation in D, the green ones is changing, but the red ones are not. So that brings us to this point that the cells are really resistant to enzalutamide. So only one gene overexpressed, Tribbles 2 has the power to make the cells resistant. Now the third part is about if Tribbles 2 works, then how it works. So we want to know the mechanism. Without mechanism, nobody is going to believe it. So we need some documentation in terms of experimental evidence, like, well, look, this is what is happening. In some cases, ERPC develops because of androgen receptor reactivation, meaning somehow the cells managed by mutation or a deletion or something overexpression, they have androgen receptor function, they regain the power of androgen receptor signaling and they become resistant. So initially our idea was, let us see what is happening to the androgen receptor. Now what you can see here, that to our surprise, we did not see any change. We did not see any increase of androgen receptor in our gene transfected cells. So meaning it is not increasing. Kind of difficult without a mouse. Anyway, so you can see that AR level is not going up or not going down, not going up, not remaining the same. It is going down. So that brought us another twist, like, okay, if it is not AR, then what is happening here? I told my fellow, can I have a plate, hold on a microscope? Now you can see that you can see the cells, the Tribbles to transfected cells are somewhat smaller and very long processes, very spiky. That man immediately reminded me, like, well, yes, the cells getting neuroendocrine. And that happens to be true. So at the bottom, you can see all these, so we got some antibodies over there, neuroendocrine markers. Adic synaptophysin, SYP, going up. Neuron-specific enolase going up. Chromogranin A going up. PRN2, SOX2, those are neuronal transcription factors. They drive neuroendocrine characteristics, they're also going up. So in A, you see the Western blot. B is the microscope. C is the immunocytochemistry. So the transfected cells, you can see in C, the LN-cap TRIP2 cells has a lot of Tribbles too, and they are all over the cells. They are both in nucleus and in cytosol. Now, what they are doing in nucleus, we don't know yet. We are studying, like, what is the purpose of Tribbles 2 going into the nucleus. AR level going down on the right side, and BRN2, SOX2, they are also going up along with Tribbles 2. They are downstream markers of Tribbles 2. So that's what our finding initially. The other way to prove the same point is knock down Tribbles 2 in these transfected cells. So that's what you did in here. You have the resistant cell on the left side, and then Tribbles 2 shRNA, you knock down Tribbles 2. Now all these markers, BRN2, NSC, chromogranin, you can see all of them are low, but AR level is now going up. So what eventually found, the Tribbles 2 suppresses AR activity. Actually, it degrades. We don't know still the mechanism, why it binds, how it degrades, but we have some idea. Maybe that we'll be able to understand within a year. So Tribbles 2 is a driver by itself for neuroendocrine differentiation. So up to this is the summary of this part of the talk. What do you have? Tribbles 2 gets overexpressed by some mechanism. You can do a drug treatment resistant, Tribbles 2 goes up. You transfer the Tribbles 2 gene itself, Tribbles 2 goes up. Now the characteristics of those cells changes towards some of the markers, AKT, MAP-kinase, B cell is going up. Those are known for enhancing growth, invasion, and drug resistance to some extent. But the major changes that we found that these cells are undergoing neuroendocrine differentiation. Now, prostate is our model, but not necessarily we are doing work only on prostate. We found, there is very recent data, that Tribbles 2 is also overexpressed in not only ERPC or neuroendocrine, pancreatic neuroendocrine cancer, small cell lung, triple negative breast, kidney, melanoma, the list is getting bigger. So we have a bigger business, looks like. So maybe in a year or two, we'll have a much better understanding about the disease, maybe a target, maybe some drug. So the fourth part, I'll tell you that we took a chance to develop some compounds to attack Tribbles 2. So novel inhibitor, saying novel is very easy for me because whatever you find is novel because nothing exists. So you can pick up a compound somewhere, but you'll have to show documentation that it really binds to Tribbles 2 or inhibits Tribbles 2 in some way. So getting a novel inhibitor is easy, but making an inhibitor is not easy against Tribbles 2 because it is a pseudokinase, it is not an enzyme. So you cannot have a quick and easy assay method to make to find lead drugs because you cannot measure the product or substrate because it doesn't have the kinase activity. It is not a GPCR, so you cannot measure the second messenger to make it a high throughput. But somehow we managed to make a fusion protein. We fused human Tribbles 2 gene with a luciferase gene from an insect together, making one big fusion protein. So the idea was a compound that we don't know that inhibits Tribbles 2 luciferase, the fusion protein enzymatic activity, but not only luciferase will be by derivation a Tribbles 2 binding inhibitor. That's a wild guess. It needs to be tested and confirmed, actually validated by further experiment. So doing that, using that, we screened hundreds, actually thousands of compounds, all wet lab experiment. Adding drug 10 micromolar, waiting for 24 hours, and then measuring luciferase activity. But my students, they did it. On the right side, what you're seeing, about 100 compounds. I can see the one drug name at the bottom right. It's called declatasvir. This is the structure. Now, declatasvir is a anti-hepatitis C viral drug. So, meaning it blocks the multiplication, actually RNA replication machinery of hepatitis C virus. So, it's a hepatitis C treatment drug, antiviral drug. We found the first time that it is an anti-cancer activity. So that was good. So we were excited about this compound. We had to do many different experiments to prove like, well, this drug binds with Tribbles 2, yes or no? The answer is yes. You can see this is the model. Declatasvir binds with Tribbles 2, so that's one, and this is the model. Now, in vivo, we thought that we implanted a tumor, and then given the drug, declatasvir, dose dependently, there is a good amount of inhibition of the tumor growth. So up to this point, we thought that, well, we have a good drug we can use for some of the cancer treatment where Tribbles 2 is overexpressed. So the drug was, the patent was applied as function patient. The drug is known is owned by Bristol Myers Squibb. But we got the function patent, so the Heniford got, my employer, they got a patent on it. The patent is approved and is given to a company in Australia for clinical trials, and they're getting good response. Not very good, but good, some good result. So the take-home message from this talk is, we have a problem because ERPC is the prime cause for most of the prostate cancer-related deaths. So we are dealing with a very lethal form of cancer. Molecular basis for ERPC poorly understood, so a mechanism-based effective good drugs are not really available. Now the solution, we found that Tribbles 2 is overexpressed, and Tribbles 2 confers drug resistance. And in DCV, declatasvir, a pre-approved antiviral, can degrade Tribbles 2 protein and kill cells. So that's what we want actually. The patients are waiting, dying in front, and they're desperately looking for a treatment. So the potential application of this work, what we are doing, that we have ERPC treatment can be developed by targeting Tribbles 2 with drugs like DCT. So I should stop here. We have a drug development pipeline now, more coming, and I'll talk maybe in the next World Congress. So you'll have to wait until that, maybe a year or 2 years from now. These are the things. This is my team of workers. Actually, I have collaboration with many different people. The Heniford Health System, I have a big group, is getting bigger. Harvard, Dr. Beltran, Liverpool, Dr. Ayers, Dr. Link in Madrid, Dr. Josie Alumkal in University of Michigan, Vancouver Prostate Center, Dr. Zubaydi and her team, MSU Michigan State University, Dr. Elsewhere. I have grants with many of the collaborators. And I also acknowledge financial support from my employer, Heniford Health System, and NIH R01, DOD. I have 5 DOD grants so far, and a foundation called Dixter. So thank you. I'll stop here and then participate in discussion. If you have any comments, suggestions, thank you very much for your attention.